First up is Brian Blanchard. Brian is a partner with Scotian Halibut, which is one of Canada's foremost marine hatcheries. Been with the company for quite some time, and we've had the privilege of knowing and working with Brian for many, many years. And Brian's going to talk about how it is that genomics is important to Scotian Halibut. So over to you, Brian.
Thank you very much, Steve. Hello, everyone. I was doing fine until I had to follow that video. I have no idea how I'm going to make it as exciting as she did. We're just doing a very small piece of work here in Atlantic Canada. I'm with Scotian Halibut. I've been the general manager for over 12 years, and we've been involved in start to finish bringing along a technology and a business in a small community in Nova Scotia, in Clark's Harbour and in Woods Harbour. And it's been a lot of work and a lot of difficulty. And one of the shining stars of why we're where we are today is because of the genomics project and our Plurigene project that we did in conjunction with Genome Atlantic, Genome Canada, and Genome Spain. I don't have a whole lot of time, so I was going to cover a brief look on broodstock development. And I put up this as a timeline, and if you look back, it actually even goes further than the 1997. It really goes back into the early 1990s. But these are some of the accomplishments that we worked on to make broodstock reliability in our hatchery more effective. And photothermal adjustment, basically we were able to change and manipulate light regimes and temperature regimes so that we could spawn year round. We also looked at using implants, hormonal implants, to help time spawning with the females. We also, a project that looked at developing all-female populations because females grow faster than males. That work was going on. Also, egg assessment work when we started using very finite diagnostic tools to figure out lipid composition in eggs to see if we can improve through nutrition. We also that rolled into the nutrition aspect of how do we manipulate or improve egg quality through nutrition means. And then we moved into the Genome Canada work, the Plurigene project. And what that did is it really gave us a huge new tool or toolbox of information that we could apply to the breeding program. And if we drop down to the bottom where we have the broodstock and bio security and domestication program, we started that in 2004. And if you see, I'm trying to represent it as best we can. It was a set program, very linear, and it was based on traditional breeding methods. With the Genome Canada work and the linkage development and the map, we were basically able to expand that program phenomenally, and that we can basically start taking traditional breeding programs and turning them into much more tangible science-based breeding programs. To bring a female halibut along to maturity is actually a 12 to 15 year process. So if you make mistakes, we can't go back in time and fix them, and I'm not going to be around long enough to correct it going forward. So the real tool that we have that's making a difference is the genetic markers and the genetic tools that are available to us. I put this one, I love this slide. These fish are all brothers and sisters from a cohort from 2004. And the fish that's circled, if you look at that fish compared to all the rest of them, there's a reason why that fish is larger. We don't know exactly why it is, but locked in its genes is the information that we need from the breeding program to be able to best pick these fish going forward so that we're not making mistakes. We don't have the luxury of time as a private company to make mistakes. And in fact, if we had known that it was a 12 to 15 year time period to bring a fish along, there's not an investor in Canada or the world that would have supported any of the work that we've done. And so we've kind of snuck through over time, and hindsight is great, but this is the reality. And it's not only for our species, it's actually for most species, except the generation times. When we look at salmon, we have to remember that DFO and hatcheries were going on in this country for easily 100 years. And because of that 100-year history, domestication had already been worked into the population. All of the fish that we're working with were collected from the wild in the mid-90s or late 90s, and so we really only have one crack at getting this right, and genomics is the tool that helps us get it right. Just so that you get an idea, this is what a female halibut would look like. This would be the egg collection piece. We actually manually strip everything, so we actually have an ability to pick and choose who we're using. And I put this one up here. This is a poster that we did from the Plurigene project, and with it we were able to send out, the bottom part is the map that we created with the project, but we sent this poster out to the world. There's 3,200 copies all over the world that went through trade publications, and I've been in Spain, I've been in Norway, and I've been in Iceland, where these posters have been up on company walls. And if you look on the bottom, it's hard to see, it has all of our partners that were involved in this. And it was a tremendous, tremendous accomplishment. And what it did, and still has, is it says that Canada is the first to have gotten the genome correct, and this is the tool that we use going forward. And the rest of the world basically stood back and said, okay, you figured it out, no problem. And now we're doing partnerships, and we're working with Norwegian companies who are buying juveniles from us, and who are actually coming to us now for stock and eggs as part of our breeding program, because there's a recognition that we're 7 years to 10 years ahead of everything that they could start today. And because of that, and because of the genetic tools, we've basically positioned ourselves in Atlantic Canada in an unbelievably good place. The hard part is just keeping the working capital, keep the whole company going. But the reality is we have this 10-year lead, which is backed by science, and that science comes from the genome aspect of it. I just wanted to give you, this is a lovely slide. This is what's actually going on when we talk about why are we doing halibut. If we look at the Pacific halibut fishery, these are the quotas. The quotas are dropping like rocks. There's pressure to increase quota from the fishermen. There's a lot of pressure to reduce quota from the advisors. There's discussion about reducing the size of age of capture. The end result is 11,000 metric tons is what the current recommendation for the quota is, which means that there's nearly 20 million metric tons of product not in the marketplace, which is a monumental opportunity for Canada, a monumental opportunity for anybody who's growing halibut. And when we look at Norway, Norway has about 1,200 metric tons of harvest annually of farmed product. Canada, I can't even put a number up there because we were at about 200 metric tons. We've since declined to about 60 metric tons because of lack of working capital. 90% of our sales are actually to Norway right now. We're trying to reverse that trend, but part of it is just the growth that's going to happen. And again, I love these nighttime shots. The market for us is right next door. You know, this is what we're trying to do in Atlantic Canada. We're trying to produce a product that does no longer exist from a wild source, that's basically next door and looking for it. And so that's one of the success stories that Scotian Halibut's been able to talk about. And the reason we are where we are today, and it's not just because this is a group, it is because our breeding program has advanced better than everybody's, and we've been able to put science along with that breeding program, which basically says we really do know what we're doing with our decision making. So thank you very much.
Fantastic. Thank you, Brian. I hope as some questions popped into your mind, please jot them down, and we'll capture those once we get into the Q&A. Next up is Andy Stone. Andy is a business development officer with the NRC here in Halifax, and over the last while has been seconded on a part-time basis to Capital Health, and served in that capacity as part of that secondment as a project coordinator to help implement a mutational screening technology for lung cancer in Atlantic Canada. And I would bet that every single one of us in this room has been impacted by lung cancer, either directly or indirectly, and hence very interested to hear what Andy has to say. He's representing the dedicated efforts of a number of folks that have been involved in the project, so I'll let Andy offer the thank yous to them. And Andy, the floor is all yours.
Thanks, Steve. So I'm going to talk about a project that's happening literally down the street, the Mackenzie building. And it's specifically lung cancer, and I think it's in our vision that it's just the first of many different types of cancer. And what we're doing is enabling, we're kind of what I would call the back end of a lot of genomics research. In fact, I think we're actually bookended by genomics. The state that we're at right now, especially with lung cancer, is not a particularly pleasant one, where the success rates with a lot of the chemotherapies that are available today are between 15 and 20%. And what's happened in the last few years, though, is there's been some very promising new targeted therapies that completely are changing the game and increasing the response rates up to 60 to 75%. And furthermore, with what we're looking at doing is actually what was missing is this ability to what's called stratify the patient population and pull out the people who are actually have the mutation that can be targeted for. So what was missing was really a means of identifying, which patients have which mutations. And when you do this type of process, what we find is that actually the average goes up for pretty much all groups within this process, so that it's known now that giving a targeted therapy to somebody that doesn't have the mutation is in some cases worse than doing nothing. So by this whole process, it really enables the rational application of chemotherapies in the healthcare system. So it's definitely going to be better for the patient outcome, but we also think with a rational drug delivery approach will actually show benefits within the healthcare system itself. So when we break down lung cancer, we see that a little more than half of lung cancer patients have a known mutation. And what we've done at Capital District Health Authority with the help of NRC is to develop what we call a multiplex panel, or the ability to test for multiple mutations in one screening. So when the patient has a biopsy taken, part of the process now will include not just the typical pathology reports on, that categorize the type of cancer and the stage of cancer, but also now molecular profiling. So we're going to see, this is the front end of a game changer within this space, that patients will not only have kind of the traditional anatomical and stage of your cancer included in your diagnosis, but also kind of a molecular signature is going to have an impact on your treatment pathways, as I suggested. So what we have done is taken at CDHA the clinically actionable mutations, put those on our screening panel, and have just implemented this in a pilot phase right now, starting in September. I think we have about 55 patients that have actually gone through this pilot clinical program, and of those, quite a few have shown a targetable mutation, and instead of getting the traditional first-line therapy, we'll now immediately get the targeted therapy. So we're already having an impact on patients' lives. This project has happened quickly, and has been, for me, it's actually been surprising when people come up to me from Toronto and Vancouver and even some of the pharmaceutical companies and ask, well, how did you get this done here? And it really comes down to the team that's on the ground, and fundamentally has to be multidisciplinary. This includes surgery, medical oncology, pathology, the lab, as well as a lot of other folks behind the scenes. And the implementation of this technology within the hospital system is actually proven to be much more complicated than the technology itself. But it's the dedication of a team like this. And I won't read off everybody's names. We'll leave this up for a moment. And this also has been funded through a number of different pharmaceutical companies. And what's really interesting is for a consortium like this to work is that when you look at the different types of mutations, they tend to be mutually exclusive. So the pharmaceutical companies aren't really competing in this particular instance. You either have a mutation that would lead you towards their drug or you don't. So they're all happy to develop kind of this multiplex screening technology so as a mechanism to direct the patient towards whatever drug is most appropriate for them. And then just to finish off on why I think this is bookended by genomics applications is that the technology platforms we're employing right now really only allow us to look at a relatively small number of mutations, and we know already, we're already thinking of how are we going to supersede ourselves, and it's probably with a genomics platform probably honed in specifically on the cancer exome, so just those genes that are involved with cancer, which would reduce the amount of data by probably to 1/1000th, and looking to now screen in the future all patients for their entire cancer exome, and building a tremendous amount of data that can be mined retrospectively in a clinically annotated database. So it's quite a future, I think, ahead of this group. So, great, thank you.
Last but certainly not least, our third and final speaker is Dr. Michael Dennis. Michael is the president and CEO of Performance Genomics Incorporated, and as many of you know, was also a former president and CEO here at Genome Atlantic. So once again, someone we've known for many years. Thanks for joining us, Michael, and the floor is all yours, sir.
Steve, and thanks for the invitation. Pleasure to be here. Tell you about Performance Genomics Inc., PGI. PGI is part of a revolution that's happening in agriculture, specifically in livestock breeding. As you know, livestock breeding has been used for millennia to improve progressively the genetic performance of livestock herds. Through the quantitative analysis of adult offspring, to looking at how adult offspring perform, to be able to pick best breeders to improve the herds. The revolution that's happening now is allowing livestock breeders and producers, rather than waiting for adults to determine how well a breeder does, analyze the genome sequence of newborn livestock and using algorithms developed that can convert the genome sequence into predictions of future performance, select those animals and only those animals with high potential to raise within the herds and to produce the next generation of livestock. It's moving very quickly. It's heralding really a new age of genomics in applications, and I'm calling them genome apps. We're going to have a set of genome apps that can go in, look at a sequence, identify specific markers that have importance for specific traits, and make quantitative predictions that can be used in everyday decision making and management in livestock production. Where do we fit? Well, PGI has a unique asset, biological asset, developed over 25 years, a mouse colony that produces twice as many litters and has twice the reproductive lifespan of a control mouse. Genes in that mouse have now been selected over time to give those mice superior performance. Our objective, identify those genes in the mouse, use them to translationally into other mammalian species, specifically livestock, to identify those sequence differences that are responsible for performance differences, specifically within the area of reproduction. So our objective, a genome map to predict a reproductive lifespan of livestock and to allow dairy producers or livestock producers to progressively improve by selection the reproductive lifespan. This revolution is being driven. We've heard a couple of references to this revolution in sequencing that is driving really the practical applications now of genomics into industrial life and in agriculture. The sequencing explosion, Steve referred to the $3 billion and 10 years that it took to do the first human genome. In the period, the 12 years since, a genome has come down to under $5,000. We see $1,000 here from $3 billion, and we're talking about 2 weeks instead of 10 years. Million-fold reduction in cost, hundreds of times faster, more powerful, much cheaper. So the application is being driven by reducing costs. It's possible for a dairy farmer, for example, to test his cow for $40 now for a specific set of markers that allow him to select the cow for better traits. The other thing, obviously, is that reduced sequence is allowing scientists, the industry and the academia, to sequence essentially every commercially relevant species, the genome of every species out there. Humans started 2012, chickens followed, mouse and cattle in 2009, 2 weeks ago swine. And sheep will be next year, and goats will be the year after, and we can expect in 5 to 10 years, all of the commercially relevant species will have full genome sequenced. They're sequencing genome of Nova Scotia mink at the agricultural faculty in Truro right now because it's an important species for the region. So that's driving really the appearance of this genomics technology within everyday life. And if I could put it in context for our target, dairy farmers have been working for hundreds of years to breed that perfect cow, which would have a good humor, stand comfortably, have long health, good fertility, obviously good milk production, but at the end of the day would be profitable. And increasing profits is still the primary driver for dairy producers. How do we increase our profits? Using traditional breeding, selective breeding, turbocharged with artificial insemination, which is 90% of dairy reproduction now is artificial insemination. It was possible to increase over a period of 30 years by 2 to 3-fold milk production per cycle per cow. So incredible success in increasing that aspect of dairy cow performance. But in parallel, other things declined because they weren't understood. Fertility in particular progressively declined. Where cows used to last 4 to 5 cycles, they now last 2 to 3. A dairy farmer with 100 cows needs to have 80 cows non-productive just to supply 40 per year to replace the ones that are culled every year. So there's a 40% cull rate. Increasing by 1 cycle, 1 more pregnancy would cut $15 billion cost out of the industry in replacements, the global industry, and add 10 to 20% to the bottom line profit for a dairy farmer in Canada. That's where we are. We have a set of, we don't have our genes in mouse identified. We have our platform in development. We're applying the latest tools to the identification of those genes. We've identified and signed an agreement with a major partner that is already out selling genomics tests for dairy cow selection. Over the next 18 to 24 months, we'll be mining our mouse and transposing that information into dairy cattle genome with the objective that within 2 years, our partner will put those tests, new tests, in the hand of farmers to allow them to select breeders that they'll end up with herds that have fewer numbers, but they'll be more profitable with fewer, healthier, longer-lasting cows, which is really a practical and very concrete application of genomics, which is already in the marketplace. So thank you.
Very well done, gentlemen. I think 3 excellent examples spread across 3 very different sectors that complement the message that was in the video, that yes, of course, genomics technologies are relevant to every sector. and virtually every aspect of our day-to-day lives. And yes, imagine this kind of stuff does happen in our own backyard. Brian, let me start with you. You've no doubt heard the projections that are out there that we're soon going to have somewhere around 9 billion mouths to feed. Some suggest that global food production needs to double in the next 50 years. Given that need for global food protein, what kind of an opportunity do you think it creates for the aquaculture sector, and how prominent will genomics be in that context?
Is this on?
Yeah,
Okay.
you're on.
Oh, that's great. Aquaculture is an important source of food going forward. Well, thanks, Steve. The reality is that there is a monumental.
Just.
Is that there now?
There you go.
Oh,
Yeah.
okay. There is a monumental shortage of food coming, and the only existing new source of protein, or the only source of protein that we've seen really over the past 25 years, has been coming from aquaculture right now. And in every country outside of Canada, growth is stable between 6 and 8%, and in this country, we're actually we're stagnant. We've basically flatlined for a decade now, mostly because of the discussion of farm versus wild, which is another topic altogether. But when we look at as we go forward, the genomics tools are really part of that breeding, that same discussion with all agricultural crops, where we're trying to increase the productivity of the animals that we're growing. In the current sector that we have, costs are constantly going up, and the only source of cost reduction and improvement in productivity is through better breeding. There's nutrition, other areas that we can work on, but really fundamentally what we're seeing is aquaculture is still new in terms of a protein source. For us as halibut, we've only touched the potential growth potential that we have. We've seen salmon is improving. Health issues can be improved with genomics, and really it's part of unlocking the toolbox of what's going on with the animals to improve them. And really Canada has a tremendous role, but again, part of it is we've got to get beyond the discussion of farm versus wild and start looking at in particular, Nova Scotia, we have 13,000 kilometers of coastal resources that really aren't being used. And if we took all the aquaculture sites in Atlantic, or in Nova Scotia, and put them together, they would be 1/10 of Bedford Basin. So it's almost criminal at this point in time that we're not trying to manage these resources properly. We have the resources, there's a demand for food, and we just need to basically move forward on that.
Great, thank you. Andy, there's an ongoing debate, and it's one that's not going to be solved for a while. Two equally prominent arguments that are out there. As genetic tools for diagnosis and patient management emerge, some argue that that ability to customize treatment and detect disease earlier will ultimately save the healthcare system significant dollars. The flip side, and those on the other side of the argument say, geez, every time you guys bring another genetic test, it ends up costing us more money. How is your group sort of beginning to explore that?
So we've built into our project a health economic assessment. So as we're running this as a pilot clinical study, we'll also be gaining information on what the financial impacts are, which are a tremendous amount of variables. Certainly, it's not all economic. When you're talking healthcare, the patient outcome is factors very prominently into any assessment. But we definitely want to understand the economic impact. And it's a little too early to say. The test itself is very cost effective. When you compare it to the cost of the chemotherapeutics, but factored in as other factors are the cost of side effects, cost of hospitalizations, the targeted therapies tend to be delivered orally as opposed to intravenously, which means they can be delivered at home as opposed to a hospital stay. So these are all the different types of factors that are going to be measured, and we're all excited to see what the results are. But irrespective, at the end of the day, this is better for the patients.
Thank you. And Michael, in your presentation, you cited some excellent examples of the potential net revenue or bottom line contribution that some of these genetics tools can make. Can you expand on that a little bit in terms of things dairy farmers can do today that even 3-5 years ago they were not able to do?
Sure. I guess impact of genomics, I put it on the two players in there. There's the breeders, the ones that sell the bull semen that is delivering the improved genetics for the dairy farmer. And then there's the dairy producer himself who has tools for the selection in his own herd. The development of bulls for artificial insemination market was cut from 5 years to 2 years, and the cost was cut about 90% by the application of genomics tests on young bulls to get as accurate a prediction of their genetic value as 5 to 6 year old bulls with thousands of daughters, which was the general way that they rated bulls for genetics. So a huge impact on the cost base for the breeders to be able to bring bulls to market. Unfortunately, it hasn't been passed on to farmers yet. Semen costs are still the same. We expect they may profit from that. That all happened within 2 years of getting cattle genome sequence done. On the dairy producer side themselves, dairy producers are slower to uptake. They need to be convinced that this new cost for them is going to give them a saving. But the combined work of the Holstein associations, Pfizer Animal Health, GeneSeq, the ones that are providing them, they are now taking up tests that allow them, again, by testing animals shortly after birth, by testing female calves for milk production, for conformational traits, for all kinds of important production traits, they can predict now how they will perform at birth rather than waiting and raising twice as many and knowing that they're going to cull half of them. So it's already having an impact, but it's still early days for the dairy producers. Probably 4%, a little less than 4% of them in the States will use them this year, but that's growing really, very rapidly.
Okay. Thank you, Michael. Any questions circling around? Kevin? Sue?
So I have one for Brian. Brian, I may have missed it. You may have said it in there, but when you're talking about bringing a female halibut to a large fecund stage, and with the picture you had up there, have you been able to select for speed or select for size in the same amount of time? Can you bring them in earlier at the same size, or do you have to still wait, but they're just bigger and therefore more productive?
It's really, is that on?
Yeah.
Yeah. It's really, because there's a nice piece tie-in to Michael's last comment as well. The females, in the wild, we have data that shows that, let's say a 30-inch halibut in the wild could be between as young as 4 years old, but as old as 12 years old. That data is just starting to come out of sort of fishery stats that's becoming easier to analyze. They're starting to put the data together. From us, we have to basically do a retrospective look at what we have, because none of these tools existed when we started. And from a female point of view, we're looking at optimizing the environment that we grow the fish in to try and speed up the process. We also cull slower growing fish, and we only pick basically the top performing. It would be nice if we could actually use a selection tool that would say, okay, at age 2 we can say we're going to keep this, and we're going to, instead of going to the 12 to 14, we're hoping to get that age down to 7 years. Part of it really for the females is a function of size. Anything less than 20 20 pounds is not reproductively mature yet. And an ideal size fish for us to work with, because the egg volumes are related directly to size, working with fish that are 70 to 100 pounds is ideal in terms of our ideal size. We've had fish as large as 280 pounds. They're just too big. You can't, they're just too big to manage in the tank. In fact, we had one fish that used to terrorize me every time I was in the tank. And she knew it. You know, and when she'd opened her mouth really was, you know, you could put your whole head in there. And, now, but on another serious note, the males, we can see reproductive maturity as early as 2 years. And when we're dealing with the males, if we have good tools, then what we can do is we can have a selection program with the males that for every one female, we can almost see maybe 5 generations of male breeding happen, because we have the tools and because we have it, and it's a trade-off of precocious male versus, you know, a male with very good genes that just reproductively active. And the males basically mature or have milt available at 1.5 kilos. So really, that's a huge difference, 3 pound versus 20 pound. So that's one of the real benefits of the genomic tool, is that we can go into the males and look for reproductive gains. Right now, most of our gains are targeted at survival, which inherently happens through survival from the first generation. In terms of growth improvement, we know that won't come until maybe F2, F3, F4. So this is where, if we target the males for that growth improvement and trying to make sure that we're picking the best female breeders, then we can try and make as few mistakes as possible. The other aspect, which was I got rushed along, so I didn't get into the cryopreservation. We basically started a program 5 years ago looking at how to store the milt for use on an ongoing basis. And for the past year, we've basically used that as a commercial tool. All our milt is cryopreserved, and it's no different than the bovine industry or the aquarium. We just take the milt when we need it, and we thaw it and use it. And those tools mean that it's available to use any time of the year whenever a female is spawning, where up until about 3 years ago, even 2 years ago, you would basically be at the mercy of whatever natural spawning was happening. And so you had no selection or management criteria with the males. And so the tools are just unbelievably available for us to make use. It's just, how do we best management? And part of the challenge for a company, for us, is how do we intellectually figure out how to do this? Because sometimes we don't have the resources to go out and hire a breeding PhD to come and work with us, even though it really is a necessity if we're going to run a breeding program. So, and I'll leave it there.
I've enjoyed your presentations. I think they're fabulous and what's going on. I'm going to direct this to Michael and to Brian, because I see Andy in a bit of a different light. When is the general public going to stop thinking of genome research as genetic altering of product?
The great question, we actually had talked about it as, what if that question comes up? But essentially, I think we come back to the straight answer. It's selective breeding. It's just a more advanced selection test that allows earlier selection and cuts costs. There are no alterations. You might have thought dairy farmers would have been people that said, you know, we can't do GMO cows. I won't be able to sell my milk. This is not GMO, is it? They had no problem. They had no problem seeing that, you know, what they're really looking at is a number of genetic potential, and that genetic potential allows them to make selection decisions.
Why are we so afraid of the word genetic altering, let's say? Why is the public, why is there that fear there that we'll eat some halibut and something will suddenly grow a third ear out of the side of our heads or something because it's being genetically altered for breeding purposes, or it's been bred for the best result versus a result that we're terrified of?
bred for the result, not altered. And it has to be the answer. But how do you address that with people? Because even GMOs, they probably have their place. They are in our supermarkets. They may not be labeled. We all eat them. We may have a fear, but we do have to overcome our fear and look at the science base of what is the policy, food policy in the country. But for us, and I think Brian, the same idea, like it's not an issue for us because we're just helping do a process that's been done for 1,000 years.
Yeah, the GMO comes up all the time, especially with the transgenic salmon. It's probably the leading, it's a technology developed in Canada, perfected in Canada, will be exploited outside of Canadian borders, will become a reality somewhere. We can't talk about it, but everybody buys Basel. Yet GMOs are bad. It's just incredible. And what it is if you can put the right title on something like Frankenfish, well then you can kill a market instantly, or you can create public perception. Salmon has its challenges because of wild salmon resources. So there was always the protection through DFO of wild stocks, which, again, if we look at what's going on in southwestern Nova Scotia, because of acid rain, there is no wild stock going to come back to any of those rivers until those rivers are rehabilitated. So everything keeps getting confused, and on one hand GMOs are bad, but again, go to the supermarket and start taking all the GMOs out, and all of a sudden people are going to be oh, I didn't know that was a GMO. And so it really is, these are market perceptions. And I guess I alluded a little bit to the rest of the world is continuing to move forward and grow fish and grow other products and do things. And in Canada, we've flatlined for a decade because we have discussions over what we think is right or wrong, or perceptions. And I don't know how to get past that. In some respects, we're all very tired of it. And it's just, it's worn us down to the point of very good work on transgenics in Canada gets exploited outside of our borders because we do great research here, but we can't commercialize things.
So how do we change public perception? Where does that have to come from?
You know what, that's strong leadership. When a comment is made like farm salmon is poison, that's when the Minister of Fisheries or the Minister of Health has to stand up and say, that's incorrect, that's a false statement. And often because of the politics of everything, we'd end up with those statements not being made. And so it's a challenge, and the leadership has to come from everybody. For us in the industry, we put in a lot of time in education. We try to do the outreach. We try to work. We have open door policies for people to come and see the work that we do. But sometimes that's not enough. And sometimes the budgets of people who have another message are significantly bigger and better than ours. And how do you compete with a good marketing campaign if you're a farmer and you're trying to stay in business because your costs are constantly going up? And that's where, for us at the aquaculture level, our associations are important. But it's also important that everybody that's involved in the sectors come and sees what's going on and becomes informed themselves. Because as soon as the facts come through, we've never had a problem. Because the facts and science for us are fantastic, because that allows us to go forward.
Well, we hit 35 billion today in Canada, so I'm kind of hoping it happens soon.
So do we.
Any others at the back?
So is there some interest in maintaining some biodiversity, or is there a thought that selecting some fish will still have enough biodiversity so that if there's an insult that eventually occurs and attacks that particular genetic species, that it may not be an issue? Is that thought to be an issue or not? When you're selecting fish or selecting for any particular gene species?
Well, it's interesting because the breeding program, and this is where we have to, today we were talking about breeding program. The breeding program is actually selecting for biodiversity, and we're continually trying to add to it from outside sources and new sources. In fact, we're looking at new wild sources to bring in as well. But that needs to be not confused with production strategies, where we're trying to pick, so we have breeders, we have multipliers, and we have production. And really within the organization, to base a production on one selected very narrow line would really just, it doesn't work. It doesn't work in traditional programs. And so the breeding and that's part of the huge cost of the breeding program, is that you're trying to expand it constantly, but the production needs is looking for high-performing fish or high-performing animals. And so it's the extra burden of making sure that biodiversity is in there that makes it challenging for us. And in fact, if we could bring in stocks from other countries, we would. Unfortunately, there's geopolitical borders that doesn't allow transfer of fish into our programs. That would, if we brought in fish from Norway to try and expand that biodiversity, we wouldn't be able to actually sell anything, because none of them would be allowed to go into Canadian waters, because it would be considered a foreign species. You know, even though Atlantic halibut live in the Atlantic.
Maybe just one or two final questions, and then we'll let everyone get back to enjoying some networking and some refreshments. Any others from the audience? Maybe I'll just ask the last one and direct it to Andy. And one for Michael as well. One last question for you in terms of how rapidly technology is advancing. No doubt the diagnostics and the tools we're using to stratify treatment are going to need to advance just as quickly. So are there things in play with your group and collaborations and partnerships that are in play that you think can help your group stay right at the cutting edge?
Yeah, I think it's key, and you talked about the advantages of being big and being small, and I think when you're small, you actually need to tap into some of the bigger players who are really at the forefront of science, and we've done so. There's a group in the U.S. It's a consortium on lung cancer stratification, and we've tapped in through both Vanderbilt and Mass General, and contacts with other folks to make sure that we're up to speed on what they're doing, because we're simply don't have the research capacity that some of these other centers do. I think we've proven ourselves in the implementation side, but we definitely need to collaborate with some of the larger centers to stay on current.
Okay. Michael, I'll give you the last word. Obviously, your presentation was focused on dairy cattle, but if I remember correctly, in some of the earlier days of PGI, they were targeting the swine industry. So just wondering if you could make a comment about, in general, how these kinds of technologies are being used in other livestock species.
For livestock, there's a general rule now established with Holstein cattle, the number of stages to go through to end up with a genomic selection tool. So they will all have their 50,000 SNP or 10,000 SNP screens developed specifically for each species. They'll be used in research projects where they relate the pattern of the markers with the specific traits that they're observing, the phenotypes in the target population that they want to select. So each one of these is going to have its genome app. All livestock breeding involves replacing breeding females when they run out. That replacement cost is the second largest after feed across livestock production. So we see the opportunity for a mammalian marker panel that would be adapted depending on the species or the breed, that would sit on top of the existing panels and improve predictions for reproduction. So we see it as a way really to move into those other livestock species as they start to adopt genomics selection as standard procedure. There was a period as well that PGI was very focused on applying this technology for humans. The idea was women early in their careers would be able to determine, do I have a long window or a short window in order to have my childbearing? Should I make a decision now to stop working and have my kids, or should I wait till can I wait till I'm 30? It's still on the back burner. We've put it on the back burner. We've heard up yes and no. Does it make any sense? But clearly, genes that control fertility and reproductive longevity in mammals have an application in human. How they get applied, whether it's in a specific health arena, remains to be seen, but that's for down the road.
Well, we promise to get everyone back to networking as quickly as we can. We had 3 excellent presentations in 3 very different sectors, all of which are end-user needs that are being addressed through genomic technologies, which is our focus and certainly articulated in the video. So please join me in thanking Brian, Andy, and Michael.